Welcome to the lecture series in food biotechnology. In the last introductory lecture, we saw how the understanding of fermentation paved the way for the development of a large number of products. In today's lecture, we are going to see how the discovery of fermentation, the understanding of the bacterial metabolic pathways, how that has been used by the industry, not only food, but all the industries to develop products utilizing these microbes. This is including the food industry. Fermentation has been known and practiced by the humankind since prehistoric times, long before the scientific principles ever understood. For example, if you consider the production of bread, beer, vinegar, yogurt, cheese, wine, all were well established technologies in ancient Egypt. This picture shows bread making on the wall of an Egyptian tomb. This was dated 1400 BC, which means that the discovery that the knowledge about bread making was known and that fermentation is required was known even then. This is grape kneading to produce wines. This was also known in Thebes. This picture is on the walls of Nakhet's tomb in 1400 BC. This also indicates that the knowledge of fermentation was known in the ancient days. Many microorganisms can grow on grape sugars more readily and efficiently than yeast, but a few can withstand the osmotic pressure raising from the high sugar concentrations. See, when you take the grape juice and allow it to ferment, the sugars in the grape juice will get converted, will get fermented, and you have a lot of microbes which can grow in them. But the point is, and you have some yeast also which grow there. The microbes can efficiently ferment, more efficiently ferment than yeast. But the problem with the microbes is, as the fermentation happens, the pH will change and the osmotic pressure will change, and very few microbes can withstand that change. Also, as the sugar is fermented, when the alcohol fermentation concentration increases, only osmotolerant, alcohol tolerant organisms would be able to grow then. The scientific understanding of fermentation microbiology and in turn biotechnology only began in the early 1850s, after Louis Pasteur had succeeded in isolating two different forms of amyl alcohol, one of which was L form, optically active, levorotatory, while the other one was not. So, Louis Pasteur discovered two types amyl alcohol. One was levorotatory, which was dextrorotatory, and the optically inactive form resisted all Pasteur's attempt to resolve it into its two main isomers. So, the inactive form could not be converted into a combination of the dextrorotatory and levorotatory in spite of Pasteur trying his level best to do it. This led Pasteur into the study of fermentation to find out the underlying reasons behind this observation, why you had an active form and why you had an inactive form, and why this inactive form was incapable of getting broken down into the active and inactive form. That was his question. In 1857, Pasteur published the results of his studies that fermentation is associated with the life and structural integrity of the yeast cells rather than their death and decay. So, the concept that it is the live cells which during the course of their metabolism will utilize the sugars which are present and do the fermentation and produce alcohol. This was Pasteur's observation, that it is not the death and decay of the organisms, but rather the live and the structurally metabolically active cells which do this fermentation. He reiterated the view that the yeast cell is a living organism and that fermentation process is essential for the reproduction as well as the survival of the cell. The publication of this classic paper marks the birth of fermentation microbiology and biotechnology as a new scientific discipline, that it is not death and decay associated with a microorganism, but rather the life, the replication and the metabolic activity which is associated with fermentation. This was actually a breakthrough paper, a classic paper. The second stage in the development of fermentation microbiology and biotechnology began in 1877. Moritz Straub proposed that fermentation and other chemical reactions are catalyzed by protein-like substances, and these substances remain unchanged at the end of reaction. So, for causing fermentation and the other related chemical reactions, you required protein-like substances. The term enzyme was coined later. So, what we are now talking about is the protein-like substances which cause the fermentation. And he also said that these enzymes remain unchanged at the end of the reaction, what we presently call as the catalytic activity, the enzyme remaining unchanged, but the reaction proceeding. He described fermentation as a sequence of events in which oxygen is transferred from one part of the sugar molecule to another part, culminating in the formation of a highly oxidized product, that is carbon dioxide, and a highly reduced product, that is the alcohol. In 1897, Edward Buckner discovered that sucrose could be fermented into alcohol by yeast cell-free extracts, and he coined the term zymase to describe the enzyme that catalyzes the conversion. So, the knowledge of zymase came in 1897, and for this discovery he received the Nobel Prize in 1907 for his research on zymase and the discovery of the cell-free fermentation. Then came the outbreak of the First World War. The outbreak of the First World War provided an impetus and a challenge to produce certain chemicals. There was need for the glycerol, an essential component in the manufacture of ammunition, because no vegetable oils could be imported due to the naval blockade. German biochemists and engineers were able to adapt yeast fermentation, turning sugars into glycerol rather than alcohol as soon as the war was over, because the glycerol could be made very cheaply as a byproduct of the soap industry. So, after the World War there was no requirement for ammunition, but during the World War they required more amount of glycerol for the ammunition. So, they had to adapt the yeast fermentation. Instead of converting it into alcohol, it had to be converted into glycerol. Fermentation product had to be glycerol, and as the World War ended, this actually lessened, because the need was lessened. They could now convert back. The diversion of the carbon flow from alcohol production to glycerol formation was achieved by adding sodium bisulfite, which reacts with acetaldehyde to give an adduct that cannot be converted into alcohol. During this process, NADH accumulates intracellularly, thus perturbing the actual steady state redox balance, that is the NAD-NADH ratio of the cell. The drop in the intracellular level of NAD plus is accompanied by a sharp drop in the flux through glyceraldehyde 3-phosphate dehydrogenase, which in turn allows the accumulation of the two isomeric forms of triose phosphate, that is glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. Accumulations of the latter along with the high intracellular levels of NADH trigger the expression of the glycerol 3-phosphate dehydrogenase. This in turn will divert the carbon flux from ethanol formation to glycerol formation. Thereby, the redox balance would be restored within the cells by regenerating the NAD plus. So, successfully the cell could be prevented from making alcohol and divert the pathway to produce glycerol. Following the First World War, research into yeast fermentation was influenced by the work of Carl Neuberg and his proposed scheme, which was a biochemical pathway for the conversion of sugars to alcohol, that is the typical alcohol fermentation pathway. Neuberg's work gave impetus into research on the oxidation reduction mechanisms and the kinetics of product formation in a wide range of enzyme catalyzed reactions. Such studies were important to the development of modern biochemistry as well as fermentation biotechnology. When the conditions of fermentation change, the enzymatic activity will change. This change in the enzymatic activity would in turn be expressed in the quantity or the amount of the product which is formed. So, to stabilize these, how the different conditions affect the enzymatic activity and how this in turn will result in the conversion into alcohol or in the conversion into glycerol. So, what are the pathways which are involved and which are the checkpoints there. So, all these studies were very important to the development of not only modern biochemistry, but also fermentation biotechnology per se. This was followed by the discovery of penicillin in 1920s, and the antibacterial properties of the penicillin were also discovered. In 1940s, this represents a landmark in the development of fermentation biotechnology. And the discovery of penicillin, which is an antibiotic, was a boon to a country in war, and it was a sensational and an invaluable discovery. And as you know, this discovery was accidental. They saw that when when Fleming was growing the organisms in a Petri plate, he found that certain yeast cells or spores germinated, and in those areas the bacteria died, the cells colonies died, and then he tried to discover what was the activity caused by these organisms, and by this was the discovery by the discovery of Penicillium notatum. So, the growth of this Penicillium notatum, the producer of penicillin, it was found to be susceptible to contamination by other organisms. So, when you talk about an industrial production, as we have dealt earlier, you require a pure culture of the organism, and you require the pure culture to produce a large amount of the product, then only it can be commercially viable. But Penicillium notatum, which was first discovered to be the producer of penicillin, it was highly susceptible, it was very sensitive, and it got contaminated with other strains, and therefore, you had to follow stringent aseptic conditions for the production of penicillin. Okay. So, this actually led to the development of what we call, so from the Petri plate to the small flasks, this led to the development of the stirred tank bioreactors, which minimize the contamination with unwanted organisms. The demand for penicillin prompted a worldwide screen for alternative penicillin producing strains. So, since Penicillium notatum was being very sensitive and it was very difficult to maintain the aseptic conditions for it to produce, people started searching for other strains of the fungus which could produce the same antibiotic penicillin. So, this led to the discovery of Penicillium chrysogenum, and Penicillium chrysogenum actually produced more penicillin than Penicillium notatum. Then once this organism was identified, this organism was rigorously tested into by random mutagenesis, very intensive program. Each mutant was studied for its various properties, including the production of penicillin. And the mutants which showed high levels of penicillin production, they were selected, they were chosen. Then these mutants were further subjected to further rounds of mutagenesis. So, by subsequent rounds of mutagenesis, they could finally arrive into one specific strain of Penicillium chrysogenum, which showed massive increase in the production of penicillin from the initial 1 gram per liter to more than 20 grams per liter of the culture. So, once the antibacterial spectrum of penicillin was determined, and it was found to be far from universal, meaning penicillium can work as an antibiotic for many organisms, but it is not broad spectrum extreme, and there are organisms which are resistant to penicillin or where penicillin does not work. So, you now needed to search for other antibiotics also, and the probable source was like penicillium from Penicillium notatum or Penicillium chrysogenum. You could get this as from the metabolite from prokaryotes. So, you could, using prokaryotes, you could obtain substances with antimicrobial activity. These screening programs led to the discovery of many antibacterial agents produced by various members of actinomycetes. The next stage in the development of fermentation biotechnology was dominated by the use of regulatory control mechanisms for the production of amino acids, meaning you know the strain, you know that it will produce this. So, now what you want to do is, you want to know the optimum substrate, you want to know the optimum pH, you want to know the temperature, you can vary all these conditions and make a regulatory issue saying that if this is the condition under which this organism is grown, I am going to get this much production of the product concerned. This was used for the first time in the regulatory control mechanism for the production of amino acids. The first breakthrough was the discovery of glutamic acid overproduction by Corynebacterium glutamicum. This discovery was happened in the late 1950s, early 1960s. This was hampered by the induction of the degradative enzymes once the extracellular concentration of the amino acid increased beyond a certain level. Glutamic acid was overproduced, but the point was, once inside the cellular cytoplasm, the quantity of the enzyme produced reached a threshold level, Then that in turn triggered other degradative enzymes which came and reduced, which could degrade the enzyme produced so that its level could be contained. So, the extracellular concentration of the amino acid, when it is increased beyond a particular time, where the degradative enzymes were activated automatically in the cell. For example, accumulation of tryptophan induced the production of tryptophanase, thus initiating the breakdown of the amino acid. This problem was resolved by the use of one penicillin, which along with tryptophan as the sole source of carbon in the medium eliminated the growing cells. In the last decade, fermentation biotechnology has taken a leap forward in consequence of the rising price of the oil and international concern about global warming. Brazil was the pioneer since 1970s in developing bioethanol production from its huge sugarcane industry, and alongside it has an expanding industry for the production of ethanol utilizing cars. The production of bioethanol, or the capacity to make it, appears to have a buffering capacity against further increases in the oil prices. A valid opposition to first generation bioethanol as a sustainable biofuel on the grounds of minimal reduction of global warming and competition with food supply. The agricultural residues like the corn stover, the cereal straws, the palm oil waste, these are lignocellulosic biomass, and this could be used as a substrate for bioethanol production. Then you have the harvested factory residues like the sugarcane bagasse, the corn cobs, the wheat bran, the palm oil cake, etc. These also can provide a rich source for the production of biofuels. The cellulosic ethanol is not competitive with either the corn ethanol or the wheat bioethanol, let alone the common sugarcane bioethanol. But when we use hemicelluloses, these hemicelluloses are easily hydrolyzed to a mixture of the C5 and C6 sugars, and most of these cannot be fermented by yeast. So, many microorganisms can ferment these sugars, the C5 and the C6 sugars, but they produce lactic acid, not ethanol. Therefore, much effort has been gone into genetic manipulation to convert or to divert the carbon flux and stop the organisms from producing lactic acid, rather produce ethanol to be used as a bioethanol. Thermophilic geobacilli was found naturally on compost heaps, and this is also seen on the silage. This can be engineered to produce ethanol from hemicellulosic sugars with yields equivalent to those of yeast fermentation of the starch sugars. You can also have new biochemicals which can emerge out from processing biomass into biorefineries. For example, sugarcane, it can give sugar or it can give ethanol from the juice, ethanol from the hemicellulosic pith. Waxes can be produced from the external rind, plus heat and electricity from efficient combustion of the lignocellulosic fibers. So, one source sugarcane, from that you could get multiple products depending upon which is the part we are talking about. Such biorefineries would be self-contained units built close to the increasing food processing plants such as the sugar refineries, flour mills and the oil processing plants. This picture shows the diversion of the carbon flux from alcohol production to glycerol formation in the yeast Saccharomyces cerevisiae. Here we add bisulfite, as I said earlier. The functional role of the bisulfite is to arrest the acetaldehyde molecules. So, once the acetaldehyde molecules are arrested, the regeneration of NAD plus would become redundant. So, and to redress the redox balance, Saccharomyces cerevisiae will now divert the carbon flow towards the reduction of dihydroxyacetone phosphate, and it will be converted into glycerol 3 phosphate. Thereby, this conversion in turn will release NAD plus, much needed NAD plus will be released, and this glycerol 3 phosphate thus generated, then it becomes dephosphorylated to become glycerol. So, this is how we change the carbon flow from alcohol production to glycerol formation. The next part in the development of fermentation biotechnology in the later part of 1960s was single cell protein, the production of single cell protein from petroleum or natural gas, and this became a huge market, and the so-called underdeveloped countries and even the developing countries wanted to go for this production of single cell proteins. And but the problem is the development of the single cell protein died in infancy, mainly because basically single cell protein from petroleum or natural gas, mainly because the sharp increase in the price of the oil. Microorganisms play a central role in the production of a wide range of primary metabolites and secondary metabolites, industrial chemicals, enzymes and antibiotics. The diversity of the fermentation processes may be attributed to many factors. One is the high surface to volume ratio, the ability to utilize a spectrum of carbon and nitrogen resources. The high surface to volume ratio supports a very high rate of metabolic turnover. For example, if you consider Saccharomyces cerevisiae, it is able to synthesize proteins by several orders of magnitude faster than plants. The ability of the microorganisms to adapt to different metabolic environments also make them capable of utilizing inexpensive renewable resources such as waste and waste products, byproducts of farming, petrochemical industries. These can be used as a primary carbon source, and then the organism can convert it into whatever is the end product it wants. And the industrially important microorganisms include the bacteria, the yeast, the molds and the actinomycetes. If you look into the fermentation pathways of microbes, the pyruvate can be processed by multiple pathways by the prokaryotes, whereas this is distinct because eukaryotes do not have such distinct and multiple pathways. This diversity, that the multiple pathways which are being evolved to process the pyruvate, has been exploited in fermentation biotechnology for the production of fine chemicals, organic solvents and dairy products. So, if you look into this picture, what you are seeing is this is the pyruvic acid which is in the center point. It can be by homolactic fermentation, it can be converted into lactic acid, it can be converted into butanediol and carbon dioxide. By propionic acid fermentation, it can convert it into propionic acid, acetic acid and carbon dioxide. By mixed fermentation, you have the acetic acid, succinic acid, ethyl alcohol, carbon dioxide and water to be produced as a production, butyric acid, butanediol, isopropene alcohol, carbon dioxide etc. in this pathway, then ethyl alcohol, carbon dioxide in the alcoholic pathway. So, multiple pathways can give rise to different types of products. In addition to the carbon, nitrogen, phosphate, potassium, sulfur ion and magnesium, the chemical input required for the growth also includes trace elements. The necessity of such a multitude of inputs is very important, as it is required for the enzymatic activities, and each additive has a specific contribution to biomass and the product formation. So, you quantify what you are adding, then find out how it is influencing the end product. So, this way each and every component, its role can be analyzed, then we can arrive into an optimum condition with all these components in the optimum level, so as to increase the production of the concerned primary metabolite or the secondary metabolite. The rate of product formation in a given industrial process is directly related to the rate of biomass formation, which in turn is influenced directly or indirectly by a whole of different environmental factors like oxygen supply, pH, temperature and accumulation of inhibitory intermediates. So, in an industrial process, when you are culturing the microbes, the amount of the product which is formed is directly or indirectly related to different conditions like the pH or oxygen supply, whether the organism is aerobic or anaerobic or facultative. Then does the reactor have the conditions necessary for its growth? What is the pH? What is the temperature? And then do you have inhibitors which get accumulated in the cytoplasm, which will hinder the production of this biomass? The medium used to grow microorganism is critical because it can determine the level of microbial growth and product formation. In order to maximize competitiveness, however, crude materials which cost less and others are used as sources of carbon, nitrogen and phosphorus. Crude plant hydrolysis are often used as a complex source for carbon, nitrogen and growth factors. Byproducts from the brewing industry frequently are employed because of their lower cost and the greater availability. And the other useful carbon sources include molasses and whey, which are the byproducts from cheese manufacture. The next condition which has to be taken into consideration is the level of minerals and the growth factors which are critical for the medium, and this has to be formulated. For example, biotin and thymine influence the biosynthetic reactions. So, this by controlling the level of biotin and thymine in the medium, you can actually control product accumulation in many fermentations. Then you can design the medium in such a way so that the carbon, nitrogen, phosphorus, iron or the specific growth factor will become limiting, meaning after a particular time you are not going to replenish the medium with a particular source. So, then but you will still allow the fermentation to proceed. So, in that such a case, the limitation is often causes the shift from growth to the production of the desired metabolites. In a nutshell, we saw how fermentation can be utilized and how the microbial pathways can be changed and the metabolic flux converted in such a way to give the end product, a changed end product from product A to product B. And we also saw that for the industrial level production of such amino acids, primary metabolites or secondary metabolites, we require to control factors like the minerals, the type of the medium, then whether it is a pure culture, then you have the concentration of the enzymes which are involved in the biosynthetic functions. So, when you formulate such an ideal condition, then you will probably can develop a strain and make it grow on a particular substrate, giving you the maximum production of the product which you are looking for. Now, the designing of the fermenter as well as the various parameters which have to be adjusted for the production of a certain particular amino acid or a particular secondary metabolite depends upon what is the product you are talking about and what is the particular strain you are talking about. So, in each of these cases this will have to be worked out. Thank you.